So we are in the first part of the course still. We're up to the 6th lecture on mechanics of DNA, what is life? And I wanted to give you some more techniques in biotechnology. We've already discussed some measuring techniques, some separation techniques. We've gone over how to clone genes. But you might want to think about this. Are there any foods or ingredients that you've avoided or eating less of? And these days, there are all kinds of diets. Atkins this and South Beach that. Much of them have nothing to do with human nutrition, or certainly biochemistry. But you know, there's this or that fad. People are cutting carbs or cutting fats or eating proteins and things like that. And there's the results from last year. Most people said they were trying to do something. Cut back on beer or something, I don't know. Half of consumers are avoided eating some food ingredient. If you did decide yes, what were the things that you were cutting out? Sugars, carbohydrates, fat, cholesterol, animal products, cutting down on salt, spicy food. What about biotechnology products? Cutting back on your GMOs? What's a GMO? Well, most people said they were trying to cut back on fats and cholesterol. But only very few actually acknowledge biotechnology products. Are you cutting back on your genetically engineered plants? The FDA requires special labeling of food when it's produced under certain conditions. For example, only under certain conditions would it be required by the FDA to label a product. The FDA labels products, the USDA and the FDA label products by content. In other words, if you were to go to buy a can of stewed tomatoes, the first ingredient, hopefully, would be tomatoes. It's not going to tell you how those tomatoes were grown or washed or processed. It's going to tell you that those are tomatoes. Current FDA labeling does not require genetically modified organisms to be a part of that label. Organic food can be labeled as not containing genetically modified organisms. Why do I bring this up? Well, as I said, it's possible to clone any gene and move that into any organism. We've done this all over the place now, realize. Most of the corn produced in this country is genetically modified. All of the soybeans, most of the cotton, most of the canola, lots of other crops. Somebody over there was eating chips. Who was it? What was in those chips? Huh? Potatoes, was it corn oil? Corn oil, soybean oil, canola oil. There, you're eating a genetically modified product. The potatoes, probably not, but certainly the canola. Probably if you go through your grocery store shelf, it's hard to avoid eating something in your day that does not contain a product that's been made from genetically modified plants. Does that bother you? You've been doing it your whole life. And it's not labeled. Does that say that that canola has been genetically modified? No, I guarantee you it has not. So remember, we can clone any gene. I've showed you how to do it. You could do it in your garage if you were so set up. Over the weekend, in fact, it wouldn't take you long. And I've shown you that you can make artificial genes. We call these transgenes. We can move them from one organism into another, hence the prefix trans. So I could take his hemoglobin, put it behind a corn promoter, and get a corn plant to make his hemoglobin. I could do this with any gene I want. So agriculture. We've done this all over the place for traits that make sense. Large-scale agriculture has genetically modified plants for all different kinds of characteristics. I'll be getting into this in the section on agricultural biotechnology because it is controversial. Europe bans genetically modified plants. You have been eating them your whole life. That's a bit of a contradiction. Why is that? Is there a health problem? Is there an environmental issue? Why would we want to do that in the first place? But genetically modified organisms, GMOs, I can clone a gene and move it into another organism. I'll get into that specific technique when we talk about the applied parts of the class. But this has also been done in fish, moving a transgene into coho salmon, which coded for rainbow trout growth factor hormone, allowing those fish to grow faster. All of the fish shown in the picture on the right are the same age, roughly about 14 months. A company called Aqua Bounty has accomplished that, and this may aid in the production of aquaculture-grown salmon. This in itself creates a controversy. The FDA has still not approved these. Why not? Would you order genetically modified salmon? What if it were genetically modified salmon or wild salmon? What if it were genetically modified salmon produced on a farm? So tools and techniques in biology and biotechnology govern the direction over where these techniques will bring us. We have all these tools in the box now. You could say, should we use them? I could say there are good reasons to use them. Keep in mind that we're not the only organism that transfers a gene from one organism to another. Bacteria have been doing it for billions of years. So don't think that humans are so arrogant to say that, like, oh, we're really something. We can genetically modify stuff now. Well, we can do it with intent. We talked about gel electrophoresis already in its different applications and separating out molecules based on their size and charge, that molecules like DNA, RNA, and protein carry charges and allow us to separate them out in a gel according to their size. And negatively charged molecules like nucleic acids will move towards the positive pole. And we talked already about using DNA cloning to make transgenic organisms. There are all these other techniques that eventually I want to be able to explain as forming a foundation for how we work with life. If you say we shouldn't genetically engineer plants, if you don't understand how we do that, how could you decide whether or not this is okay? If it all seems like a big black box and kind of mysterious, I've done sort of Jay Leno style interviews in grocery stores. How do you feel about GMOs? Person has a basket full of them. And I say, well, then they say, well, I'm against them. Well, what are they? Well, I don't know, but I'm against them. Really? So what is it? What are the controversies? How is it done? Another reaction that I need to explain before we close out this section is called PCR, also known as the polymerase chain reaction. Remember in my cast of characters that replicate DNA, the one that was second from the left was called a polymerase, a protein that brings in an A and matches it with a T and causes that growing chain to extend in replicating DNA. That's called a polymerase. So we've borrowed that reaction from nature and learned how to develop a method that could copy DNA exquisitely, very precisely, from very small amounts. And this has had all kinds of interesting applications. For example, you leave a piece of you behind pretty much wherever you go. So this technique of being able to copy DNA has been exploited in forensic applications to decide whether or not someone is implicated or exonerated from a certain crime. Before many of you were born, President Bill Clinton had something to do with a certain intern named Monica Lewinsky. And an association was made between some material left on Monica Lewinsky's desk or dress and Bill Clinton. How did they determine that that had something to do with Bill Clinton's DNA? Well, they didn't sequence all of his DNA and her DNA and compare them. Rather, they relied on the polymerase chain reaction, which was originally invented by this man, Kary Mullis. Kary Mullis, an interesting character in his own right, and I'd refer you to this book called Dancing Naked in the Minefield, written by Kary Mullis. But he graduated with a PhD in, I think it was in biochemistry or molecular biology, and pretty much checked out of science. Decided he was going to bake bread at the Tasahara Bakery in the Haight-Ashbury district of San Francisco. Wanted nothing to do with biology or its applications ever again until someone came to him with a postdoctoral opportunity at Hoffman LaRoche saying that they had a position where he could work. And what they wanted him to look at was a certain polymerase, that enzyme that copies DNA. They had isolated it from an organism that grows in the hot springs of Yellowstone National Park, Thermophilus aquaticus. So it's a bacteria that grows at 100 degrees C. That's boiling water. So this organism is growing in boiling water. I assure you, if I put you in boiling water, you're not going to be replicating your DNA for very long. This organism is kind of happy to do that. Why? How? They isolated the polymerase from Thermophilus aquaticus. And Kary Mullis, driving home one night, jots down on a scrap piece of paper a scheme to use the heat-stable polymerase from this organism to copy DNA. This is brilliant. And if you don't think scientists are creative, check this out. And he won a Nobel Prize for this. And then again, he checked out of science to go surfing. But that's his prerogative. So imagine you have DNA that needs to be amplified. Say I wanted to isolate DNA to prove that you were here today. You've touched that armchair. You've left some cells of yours behind. You drank out of a glass. Some of the cells from your cheeks sloughed off into the glass. The knee that you're touching that chair with leaves some cells behind. All those have DNA in it. Your hat brim. So imagine you have a blood spot. So I can collect DNA from that blood spot. Now I have DNA that can be amplified. It might be all sheared up into pieces, but if I want to copy it, I have to have a target. I just can't copy it willy-nilly. I have to have some short sequence that I already know. So you need some target to be amplified. Let's go back to his hemoglobin gene. Let's say that I know some sequence fore and aft of that gene, 5' and 3' directions of the DNA. So let's say I know 8 base pairs of that. Actually, knowing the human genome now, I could simply look that up. But let's say I have a target, and I have a sample. So I can make these short primers, 8 to 13 base pairs long, of the DNA to be amplified. So the things that I need are this heat-stable DNA polymerase, the DNA to be amplified, the primers, and bases, A's, T's, G's, and C's, that I put into the reaction mix. DNA requires double-strandedness in order to replicate. So I can't just have a single strand of DNA and replicate it using this particular method. So if I separate the strands from his hemoglobin, I have 2 strands of DNA. I attach the primer that's going to recognize one end of it, and the other primer is going to recognize the other end of it. Using the polymerase, I can add in the A's, T's, G's, and C's and copy the sequence for his hemoglobin. Follow that? But then I've only made one copy. That's what his cell is doing right now anyway. So what Kary Mullis decided to do, the brilliance of this, was to use this heat-stable polymerase. So I could take DNA from a blood spot, heat it up. When you heat up DNA, it denatures into single strands. The strands come apart. If I drop the temperature down, so I heat it up to 94 degrees, and this causes strand separation. I drop the temperature back down to 55 degrees, and the primers will bind to their targets, making a specific target only on that sequence that I've designed the primers to. Then I raise the temperature back up to 72 degrees, and the polymerase does its job, copying that strand. Then I heat it back up. Now I have 2 copies of his hemoglobin gene. I heat it back up, and I've got 4 strands. I do that again, I've got 8. I do that again, and exponentially I increase the number of copies of his gene. Did you follow that? Let me just show it to you diagrammatically. 1 copy of his hemoglobin gene, heated up at 94 degrees, both templates are exposed. Bind the primers at 54, 55 degrees, they recognize their target. The heat-stable polymerase synthesizes, using free A's, T's, G's, and C's, a copy using his template. Now I've got 2 exact copies that is completed at 72 degrees, heat it back up, and they both form new templates. Do that again, and you go from 1 molecule to 4, very quickly. So this thermocycling, using this TAC polymerase, TAC, Thermophilus aquaticus polymerase, heat-stable thermocycling, allows you to go from 1 molecule to a whole blob of molecules very quickly. So I can't see one of his DNA molecules in a gel. But if I do this reaction over this number of cycles, it will form a band on a gel that I can visualize very quickly because I've amplified his piece of DNA, or yours off of your hat, or yours off of your seat. And I could prove that you were here if I amplified the correct genes. And that's how forensics is done, using that piece. But if I want to, I can copy anything. Anything. It's a method for rapidly copying DNA. And that's Kary Mullis in the 1980s did that. And that's now used in forensics, and I'll go over that again when we talk about forensic applications. But just very quickly, what this means is that each one of us is genetically unique. I can tell that. And if enough genetic variation is tested, each one of us can be uniquely identified. But do I need to sequence all of your DNA to do that? That'd be one way to do it, although very tedious. DNA is found in every single cell in your body, except for red blood cells. But there's plenty of other cells in your blood that have DNA in them. And then an evidentiary sample can be matched with DNA from a suspect to either implicate or exonerate someone in a crime. And we don't use his hemoglobin gene. Interspersed in amongst all of your DNA are these short tandem repeats, these sequences that don't code for anything, but there's a lot of variation in them. We call them STRs. These are what is used to identify people. There are unique variants in all of us. So say, for example, I wanted to identify you from you from you. There are strands of these STRs that vary in humans. He might have one that is 6 base pairs long. He might have one that's 12. He might have one that's 24. So if I have a primer that sits here and sits there, and I use PCR to copy this, I can then run it out on a gel. His being 6 is going to run at the top, being very small. 6 at the bottom, being very small, runs through it very quickly. His 12, intermediate. His 24 will be at the top. Therefore, just one of these, I can discern a marker that is not a gene that will identify him from him from him. In forensics, they use 13 standard markers. that in order to score 13 out of 13, the probability is such that there haven't been that number of people ever born in the history of the planet. So out of 13 markers like this, we can identify anyone. And the instrument to do it, that thermocycler, is very small and very accurate. So if we look at how these samples might run, the possible conclusions might be that suspect 1, suspect 2, suspect 3 were at the scene, none were at the scene, multiple suspects were at the scene. Data was inconclusive. Notice that in the case of DNA forensics, except for sexual crimes, this is all circumstantial, proving that someone was at a scene, linking someone to an object. So in this case, if we use, say, 4 fluorescent markers, we can match them, suspect 1, suspect 2, suspect 3, to an evidentiary sample, and immediately you can see the match. Let me drop those out. That suspect 1 and the evidentiary sample are the same. So it was that technology that quickly tied Bill Clinton to the blue dress. Or let's take it more current, just today's news. We learned that Trayvon Martin's DNA was not on the gun handle. He didn't leave any cells on the gun. Remember the argument that's being made? George Zimmerman says it was self-defense. Shouldn't there be some of Trayvon Martin's DNA on the gun, or at least on the holster? That was just today. So realize how sensitive and how widely used this technology has become. People are exonerated from death row, showing that they possibly could not have committed the crime that they were accused of, based on DNA evidence. This is the Biotechnology stock project. Many of you may realize that biotechnology as a field is, from a pure science, almost immediately applied. And so this is what is going to make up the second half of this course on the applications. But what we should realize is that there are entire fields and careers and industries built on these applications, whether that's in agricultural biotechnology, medical biotechnology, pharmaceuticals. There are people who make products, such as pharmaceuticals. There are people who make tools, so pharmaceutical researchers can do their work. And virtually every other supply chain function in between. So really, no matter what your field is, or what your major is right now, there are various aspects for career opportunities in biotechnology. I thought that to investigate some of this, it might be an interesting exercise to give you $100,000. That's fictitiously. I did have a student one year who didn't start this project because she was waiting for her $100,000. I don't do that. So, but you have $100,000, and the idea is to invest it in biotechnology stocks. So we will say that the first installment of this project, that's worth 5% of your grade, is outlined in the syllabus, and I'll go over that in just a minute. But the idea is basically to select 5 biotechnology companies and invest $100,000. And we can do that now, and then you will cash out, so to speak, at the end of the semester and calculate your losses and gains. So I think it'll be pretty interesting in this economy. How does biotechnology fare? If you were just to say to some relatively uninitiated investors, go ahead, invest in biotechnology. Now in your case, it's only for 3 months, but I think it's interesting to see how that might compare to, say, the S&P 500. So this is an explanation of what you can do. You can divide up your $100,000 however you see fit, but you should invest close to $100,000. I would suggest you spread that over 5 companies. You could invest a lot in one and little in another, or $20,000 each, or around there. And what you'll want to do is look up the companies. You can Google them. And I made an example here of Monsanto. They're an agricultural biotechnology company. And you can look up their stock ticker designation. For Monsanto, for example, that would be MON on the New York Stock Exchange. And you can look up what their performance has been over, say, the past year. There will always be associated with a company a stock chart or a stock history, and you can see how that stock has performed over the past year, or if you want to go back further, it might be interesting to see what they've done over the past 5 years. Some companies might be relatively new and you might not be able to do that. But you can choose your companies and calculate the number of shares that you get for the amount of money that you're going to invest. So you would want to print and submit the stock quote for each company and invest chosen amounts and calculate those shares. And we're asking you to turn in a 1-page summary of this, and I'll show you some examples in a moment. But biotechnology and industry are almost synonymous. As a field, biotechnology does embrace pure science, as I've already mentioned, but it also makes and provides and uses tools. And there are various different companies that are involved in biotechnology. Some of them might be surprising to you, as in some people that were more characteristically into other fields have realized the lucrative potential of biotechnology and life sciences. So some of them will be more familiar in terms of pharmaceutical companies, but you might want to do some investigation on that. So what we should realize is that the application of these tools that we're talking about in the biological sciences has moved from academics to the private sector. In fact, I think this might be a bold statement, but really, most of the innovative science in biotechnology right now is going on outside of academics. Now, if you look historically at sciences, this has not characteristically been true. Look in the 1940s, for example, at the development in nuclear power. It was all done by physicists and academics. And other fields were similar to that, whereas now I think what you see is a lot of the innovation is going on in industry. And this, in some ways, this is good because it's heavily funded. In other ways, it's not so good because it's behind closed doors and it's proprietary. And I will also say that nearly every innovation that occurs in biotechnology is patented. Anything that's worth patenting is patented. Similar to, you must have heard about Apple versus Samsung recently and the huge patent disagreement that they had. But this is similar to what goes on in biotechnology. At the same period of time, for example, Monsanto was in a struggle with Pioneer DuPont over their technology, and that lawsuit closed for over $1 billion. So it's not just Samsung and Apple, but this kind of patent litigation goes on in industry as well, in biotechnology. And so these innovations are driven by profits, not necessarily by need. For example, that you can see blockbuster drugs like Lipitor and Zoloft. Meanwhile, the world is still lacking a malarial vaccine. Why? How come we don't have an AIDS vaccine, but we have Viagra? I mean, it turns out that the biotechnology sector is really driven by profits and not by global need. And the distinction, as I've already made clear, between basic and applied science is really quite blurred. You will see basic science still written up in journals like Science and Nature, And there's a guy like me reading those articles. I read one last night that I think, oh, I know what I can do with that. We almost all can divide biotechnology industry into two sectors, red and green. Pharmaceuticals versus agriculture, for example. You can think of red as in blood, agriculture as in chlorophyll, and green. But there's also the information-based sciences, and biotechnology, as you'll see in today's lectures, is rapidly moving out of a descriptive science and into an information-driven science. Take that to heart, because that's really where the future is in terms of fields like genomics, proteomics, metabolomics, phenomics, bioinformatics, population genetics, and the information databasing behind clinical trials. So the role of companies is in technology development. Much of the new technology innovations come out of industry. Some come out of academics, and those are immediately patented and sucked up by companies. Hopefully, this returns investment to academics. Also, we can see that a lot of premier innovation right now in biotechnology sectors is happening in small startup companies. Take notice of that too. It very much parallels what's gone on in the information sector, as in phones and iPads. You see that small companies can take on risk that large companies can't, so that the larger pharmaceutical companies look to buy out the smaller, more innovative, risk-taking companies. And this is driven by patents. If you're going to make a small company, patents, you have to protect your intellectual property. We could get into a whole lecture alone just on patents. Can we patent human genes? Should we be able to patent genes? By the way, the answer to that is no. If you discover a use for that gene, you can patent that. And I would love to enter into a discussion with you about patents in general, especially in the United States. But I don't think we can do that in the context of this class. But certainly, we should know that these things are market-driven. There are a whole list of companies now that are just based on genomics. I will talk about this in the second part of the lecture tonight on the genomics revolution and what it means in terms of biology becoming an information-based science and what it will mean for the future of your personal health. But there are a whole list of companies that are devoted to just that. But we should also recognize that large pharmaceutical companies, Pfizer, SmithKline, etc., have their own genomics functions. So do the large agricultural companies, Monsanto, Pioneer, DuPont, Bayer, Syngenta, and others. Then there are technology companies, people that are making the tools. And some of the ads, as you can see here, I've pulled these ads out. This is not something I created. But the ads for these technology companies are really getting to be pretty outrageous. And I've included some of them in the lectures tonight that you just can't believe what you can buy these days, you know, in terms of the tools that are supporting the pharmaceutical and medical research sectors. There's a lot of money involved, and there's a lot invested in it. Whether this is robotics to move high-throughput sequencing, or whether it's making transgenic rats and mice for medical purposes. So it's been said, you know, that during the gold rush, if there's a gold rush right now on in biotechnology, it's been said, you know, that the people that made the picks and shovels during the 1800s gold rush made more money than the people who made gold or found gold. So Sears and Roebuck, for example, was founded on the premise of making picks and shovels for people to go find gold. Similarly, there are these tool makers, the picks and shovel makers of biotechnology world that are capitalizing on the growth in that sector. Look at this ad. You can buy total human RNA. You know, I'm sure you need that for your weekend coming up, but there it is from Strategene. Or Gene Jammer. If you really need to get a gene into those difficult to transform cells, maybe these are the people that you need to talk to. I mentioned that all of these technologies are patented. If you want to make a product, and I mean virtually anything from a new drug to a transgenic corn plant, this will require a whole list of intellectual property that you have to secure rights to from the patent owner and probably pay royalties to in order to make your product. For example, let's say you came up with an idea to make a new transgenic corn plant. You want to synthesize a new protein. You know you can do it. You've got the gene to do it. You've got the laboratory to do it. It'll still take you over 77 patents in order to make that. Probably about 10 years worth of your time, and probably about $136 million. So yeah, it's not something you're going to pick up as a weekend hobby in your garage. So we call it FTO, freedom to operate. So you can go to these technology companies, these biotech companies, and when you're looking up your stocks, peruse their website. You know, go to their homepage, see what they're all about. See what kind of things they're making. This is Monsanto's website. You could go into their news and what the media is recently on their website about Monsanto. You could always, you know, go and see what other people say about Monsanto, and I think that might be interesting to you as well. You can look at what their products are. What is Monsanto making? It's been said by some that Monsanto now controls world agriculture. Nearly. It might control US agriculture. Think about that. We can get into that in a later lecture. So you can see who they are, what their products are, who their investors are. Usually they have careers listed. You might want to check some of those out for you career savvy people looking ahead. What types of people do they hire? What are the criteria? For a bench scientist, you know, hiring PhDs, you might want to think about preparing yourself for a career like that. If you're in business, there are business opportunities from bachelor's to MBA levels, and virtually all types of careers in between. So don't think it's just about life sciences, but if you're in life sciences, you might want to pay attention to what an adequate resume might look like. As you know, I spent more than a dozen years in industry. I've seen thousands of resumes. I can take one in at a glance. And I assure you that my counterparts can too. So how do you prepare for that? Might be one question you might want to ask yourself. So here are some of the products that Monsanto lists, and we'll talk about these when we enter the biotechnology sector, specifically on agriculture. But here is the Monsanto stock price today, trading at $89.79. It's down 0.66 or 66 cents. And you can see by the stock chart that I've downloaded for this slide that while they had a dip in May and June, that's recovered quite nicely. And down on the bottom here, you can see what the trading for that stock is, how many shares were traded per day. So of course, you know that the old adage, buy low, sell high, that's what you're trying to do. It's hard to guess these things. Look at Facebook IPO is now worth, I think, just less than half of what it was when it was initial public offering just a few months ago. This was what Pfizer looked like today at 12.22. Pfizer is traded on the New York Stock Exchange at PFE. That's its stock ticker designation. And here you can see how Pfizer stock has performed over that same period of time. By the way, when you do this, pay attention to the numbers on the side of that graph. So it looked like Monsanto changed a great deal, and it has since last May. Whereas you can see by this graph that while Pfizer stock has gone up in that same period of time, it's only gone up by a very small margin. So some of these things you might get into for the short term, you know, bet on a penny stock and hope somebody buys it. We've had people make extraordinary amounts of money during these 3 months based on $100,000. Or you might want to think about if you were going to do this as a long-term investment, something that would be very stable and earn money over a long period of time. This is what a typical stock report might look like that you are thinking of handing in next week. So it would be due October 3rd. You want to choose your 5 biotech companies and list what the cost is. First of all, the name of the company, the ticker designation, the cost per share, how many shares you're purchasing, and what the cost is. And it can be just one page like this with those criteria, and you'll earn your 5% of your grade. Please pay attention to get that in on time. This is another example. Oh, the student's name there was Henrietta Lack. I'll get back to her in a subsequent lecture. And this fictitious guy did this with his biotech companies, but I just put this up as examples. And you can really Google biotechnology companies, and here are some that I've listed that'll be on the PowerPoint database as just some examples. The list is really quite long. Oh, sweet spontaneous earth, how often have the doting fingers of prurient philosophers pinched and poked thee? Has the naughty thumb of science prodded thy beauty? How often have religions taken thee upon thy scraggy knees, squeezing and buffeted thee that thou mightest conceive gods? But true to the comparable couch of death, thy rhythmic lover, thou answerest them only with spring. You may have heard E. Cummings in other iterations, and I'll give anybody here extra credit if they can possibly come up with the E. Cummings non-lecture series. It was 6 lectures that E. Cummings, the poet, delivered at Harvard in about, I think, 1956. I don't even think the Harvard Library has them, but if you can find them anywhere, I would encourage you to listen to those.